Good morning, students. Today's topic is phase contrast microscope. So, first of all, what is diffraction index? It is the ratio of velocity of light in air to that of any given medium. And now, what is diffraction? The process by which a beam of light is spread out as a result of passing through a narrow aperture or across an edge typically contained by interface between the waveforms produced. So, what are the advantages of phase contrast microscope? So, a small unstable specimen such as a living cell can be seen by phase contrast microscope, and it makes highly transparent objects more visible. It can examine intracellular components of living cells at relatively high resolution. Example is the dynamic mortality of mitochondria, mitotic chromosome and vacuoles, and it made it possible for biologists to study living cells and how they proliferate through cell division. So, what are the parts of phase contrast microscope? So the phase contrast microscope is a special adaptation of light microscopy and helps to obtain a clear picture of living or unstained cells. The adapters convert minute difference in phase changes in transmitted light due to refractive indices of all cell organelles into perceptible shades of gray. So this allows organelles of living cells to become visible with fair contrast in them. So what is the working principle of phase contrast microscope? So a region of different compositions likely to have different refractive indices. Normally, such differences cannot be detected by our eyes. However, PCM convert differences into differences in intensity which are visible to eye. PCM converts invisible small phase changes caused by the cell component into visible intensity changes. Phase contrast is obtained with the help of the annular diaphragm by separating the central and direct ray of the diffracted rays. The ring-shaped illuminating light that passes the condenser annulus is focused on the specimen by the condenser. So some of the illuminating light is scattered by the specimen. The remaining light is unaffected by the specimen and forms the background light. So in phase contrast microscope, the image contrast is improved by two different methods: negative phase contrast and positive phase contrast. So what is negative phase contrast? The background light is phase shifted minus 90 degrees by passing it through a phase shift ring. This eliminates the phase difference between the background and the scattered lights. To further increase contrast, the background is dimmed by a gray filter beam. In positive phase contrast, if the background light is instead phase shifting by plus 90 degrees, the background light will thus be 180 degrees out of phase relative to the scattered light. It leads to formation of an image where the foreground is darker than the background. So, how is preparation of the slide? First is growing cell, second is fixing cell, and third is sectioning specimen. So thank you, students. This is all about phase contrast microscope. In next video, we will discuss different type of microscope. If you have any kind of query, you can write down your query in comment section. I will resolve it. Thank you.